You all set, Dr. G?
Yes.
Okay, good. So good morning, everyone, and happy Friday. My name is Matangi Gopalakrishnan. I'm an assistant professor from University of Maryland. So it's my pleasure and welcome Dr. Yuan G for our KOL lecture series today. So Dr. Yuan Ji is professor of biostatistics at the University of Chicago, and his research focuses on innovative Bayesian statistical methods for translational cancer research. And he has authored over 150 publications in peer-reviewed journals, both in medical and statistical journals. And he's the inventor of many innovative Bayesian adaptive designs, such as MTPI and I3+3 designs, which have been widely used in dose-finding clinical trials worldwide. And his work on cancer genomics was reported by a large number of media outlets in 2015. He has received the Mitchell Prize in 2015 from the International Society for Bayesian Analysis, and he's an elected fellow of the American Statistical Association. So it's my pleasure to welcome him for the talk today. And he'll be talking about statistical designs for early phase oncology drug development in the era of dose optimizing for targeted therapeutics. And before I hand it over to Dr. G, just a quick request, please keep your please mute yourself during the talk. And for any questions, you can use the chat window to ask a question. And we'll have a separate question and session at the end of the session as well. So thank you and enjoy, everyone. Dr. G, it's all yours now.
Thank you so much for the nice introduction, and thanks for the DIA working group to inviting me, and welcome to this talk, everyone. And so as the title suggested, I'll probably provide a review mostly of existing designs and pointing out some novel development in the statistical designs for early phase oncology drug development, and with some emphasis on the dose optimization, which is a new initiative, at least from the FDA recently. But before I go into that topic, I will provide a review of existing dose finding designs, especially for phase 1 trials, and then using that as a motivation for the recent innovations of designs in the era of optimization, dose optimization. So without further ado, let's start. This is my conflict of interest. Just keep in mind that whatever designs or models statisticians have proposed, all of them are wrong because we really don't know what the truths are, what the mother nature is. It's usually much more complicated than our limited knowledge in terms of mathematics or statistics, but hopefully some of the models we propose or methods we propose are useful. So this is a first a slide as a review of some existing major dose finding designs in phase 1 trials in the past 30 years, 3 decades of development, mostly in statistical literature. And it's not a complete list of all the designs out there, obviously, but hopefully this captures some major developments over the years. I won't go into the details. I'm pretty sure most of you are familiar with some of the names of the designs here, like a 3+3 design, the CRM design, and then maybe you've heard about the BRM, MTPI, and more recently the Boeing design, as well as an I3+3 design that I'm going to introduce later on. On the left-hand side, it's a linear chronicle a lineage of the developmental orders of these designs. On the right-hand side here, it's more about categorizing them into different classes of designs. For example, the rule-based designs, model-based designs, including complex models or simple models. So first, just to start out, we have to talk about the 3+3 design because this is still probably the most apply the designs in practice, especially due to the reason that the clinicians are still they are very familiar with the designs. They've been using it for many years. The design have been applied in many practical trials, have been passing the reviews from the regulators as well as the IRBs. So this design was first proposed in a paper, statistical paper in 1989, although clinical society has been using it way before that time. The diagram here summarizes the 3+3 design, which is based on a pre-specified set of fixed rules. It tells you what to do when you treat 3 patients or 6 patients at a given dose. And these rules are, as I mentioned, fixed. And then based on these rules, one can design and conduct dose-finding trials. Because these rules, they actually make quite a lot of sense logically, as well as being transparent. Clinicians love them. They understand how these rules work. They mostly agree with the decisions on these rules, and therefore the 3+3 design has been very, very popular in the past many decades. So in summary, it's a rule-based design. There's no statistical models behind to drive these decision rules. It's very simple, transparent, and the clinical society already accepted as a gold standard. However, from statistical point of view, it only tells you decisions for 3 patients or 6 patients at a given dose. The targeted MTD, the maximum tolerated dose, usually has a pretty wide range under this design. It doesn't tell you what specific target is for the MTD. The performance in terms of simulation results, operating characteristics of the 3+3 design actually depends on the number of doses in the trial. The more doses you have, the a little bit worse the simulation results from the 3+3 design. There's a large variabilities at the end for MTD identification, mostly due to the fact that the 3+3 design does not allow you to treat more than 6 patients at a dose, therefore limiting the sample size for a trial. And then after the trial is finished, although the phase 1 sample size can be very small, and sometimes it's a good thing, especially on a tight budget, but you're running into a dilemma where at the end of the trial, you have often very little data to supporting later phase development, such as picking up the recommended phase 2 dose, again, due to limited sample size. So I think at this point, even in the clinical society, the attention has been shifted away from the 3+3 designs, partially thanks to a lot of folks from our society, the Statistical Society, to pointing out the issues with the 3+3 designs. These are 2 papers that are published in the clinical journals. And there are many more actually in the recent years that are also giving out the same message, which is not to use the 3+3 design. So alternatively, as a statistician, we use probability models for inference and decision makings. So in very naturally, we consider model-based designs. The probably most well-known model-based design is the CRM. BRRM is also right up there. It stands for Bayesian logistic regression method. CRM stands for the continual reassessment method. In both designs, they specify the target probability rate for the MTD, which is different from the 3+3 that doesn't specify this number. So once we have a target, then we can set up probability models trying to develop decision rules aiming to achieve or to get close to the target. Both of the designs use those response models or those response curves, such as the power curve or logistic regression. If you use Bayesian inference, then you would put prior distributions on the unknown parameters in those response curves. And the decision rules for the designs can be either be a very simple ones where you just want to minimize the distance between the estimated toxicity probability to the target toxicity probability of the MTD. By minimizing that, you are zooming into the MTD based on this inference in a consecutive fashion. BRM also sometimes use different rules where they calculate the posterior probability of certain dosing intervals like overdosing, underdosing, and target dosings. So from a statistical point of view, this makes a lot of sense because essentially what we're trying to capture is on the right-hand side here. We want to capture these dose response curve using probability models or using mathematical models. What we want to target is to find a MTD where under the which one is the toxicity curve that we achieve the maximum tolerated rate, for example, 30%, which is also denoted as PT here. And then we want to, the underlying assumption of finding such a rate or the MTD is because we assume the efficacy is also monotone with the doses. Therefore, the higher the dose, the better the response rate or the more benefits for the patients. However, we don't want to let the dose to go too high so that the toxicity becomes not tolerable. So under this regime, the statistical models and inference based on the CRM, BRM make a lot of sense. And it is how we deal with decision makings and probability inference in almost all the other problems, not just the dose finding. Operationally, these designs are much more complex than the 3+3 designs because apparently the clinicians they don't know how to perform Bayesian inference under those response curves and priors. So the trial would need a dedicated statistician to guide the inference and decision making. And also sometimes these model-based decision rules, they may not agree with the common logic from the clinicians, and therefore the safety monitoring committee sometimes need to override the decisions from these model-based inference. And also initially there are some issues with the models which produces some aggressive decisions for those findings, and therefore additional research has been proposed to rein in that, such as overdose control. So these are the model-based rules, and this was a paper published in 2019. As you can see from the title, it's a tutorial on teaching folks how to use the continual reassessment method. Keep in mind that the CRM was developed in 1990, and this was in 2019. So almost 30 years later, there still needs to be a tutorial paper to educate the society, and you can see the propagation of these complex model-based designs hasn't been super fast or quick in the community. But on the other hand, the paper is very, very nicely written, providing a lot of practical guidance on how to use and properly use the CRM for practical designs. And this is one of the really useful tables from the paper where it provides a list of software for the CRM design. You can see a lot of them are freely available, developed by researchers, and some of them are commercially available. So since 2007, there's a new wave of development trying to simplify these complex model-based designs because, as you can see, due to the complexity and the clinicians' difficulty to adapt them and understand the decision rules, there have been new research, relatively speaking, on simplify these model-based designs. So the idea is to mostly get rid of the dose response curves and also hopefully get rid of the overdose control due to the simplified modeling, and then still maintain some desirable statistical properties, such as accounting for the variabilities and still also provide simple decision tables, so for example. So let's get into that. So you probably heard about these designs. the MTPI or MTPI2 designs. The first idea from these designs is to provide or to ask users to specify not only the target probability of toxicity for the MTD, but the equivalence interval. The equivalence interval is not a parameter to be estimated. It's rather something that the user specifies, especially from the clinicians. And the idea is that you want to specify a small interval around the target, such as PT minus epsilon and PT plus epsilon. This small interval captures the potential variabilities in the definition of MTD. In other words, if we assume the MTD is the highest dose with a probability of toxicity equal to 0.25, the equivalence interval says that even if it's, for example, 0.24 or 0.26, we would still be able to consider that dose as the MTD. So by defining the equivalence interval, it already specifies the variabilities in our knowledge as upfront values for the MTD. But there's a lot of benefit once we specify the equivalence interval. For example, with the equivalence interval specification, which is here, this is the equivalence interval, then we also get the underdosing interval, which is below the equivalence interval or the overdosing interval. Then focusing on the inference of a single dose, using Bayesian models, one can get a posterior density for the probability of toxicity of that dose. Then by calculating the area under the curve for the three intervals and dividing by the length of the interval, in the 2010 and 2017 paper, we proposed to calculate something called the unit probability mass, which actually turns out to be the same as the marginal posterior probability of the intervals. By comparing these three UPMs and picking up the interval with the highest UPM, you'll be able to design a trial using up and down decisions. So for example, if the equivalence interval has the highest UPM, then the equivalence interval wins. And the associating or the corresponding decisions would be to stay at the current dose, because the equivalence interval wins means the current dose is close to the target, and therefore the decision should to stay at this dose. If the underdosing interval wins, then the corresponding decision would be to escalate. If the overdosing interval wins, the decision should be de-escalate. So by continuously apply this type of decision rules at every single current dose throughout the trial, then the trial can be realized using these simple up and down decisions. In MTPI2, rather than using three intervals, it breaks the intervals into mini intervals which have the same length. This is to overcome some features in probability inference, which is called the Occam's razor. I won't go into the details of this, but it's based on the same idea, just to calculate the UPMs under each interval or the marginal posterior probability of each interval and pick the corresponding up and down decisions for the winning interval. And that's the main idea. So the benefit of having this type of simple models behind the scene is be able to generate a decision table like this before the trial is started. So the decision table tells you in terms of up and down decisions, like escalate, stay, or de-escalate, for every possible outcomes that you can obtain from the trial. So for example, if you treat 3 patients at a dose, and if one of them has DLTs, then based on the simple up and down decisions and those probability inference I just illustrated, the decision would be to stay. And then if 2 of them have DLTs, the decision would be to de-escalate. And in the extreme of 3 patients and 3 DLTs, then you would de-escalate, and U stands for that the current dose will never be used again in the trial because it's way too toxic. So these tables can be printed out ahead of the time and be examined or assessed by the entire team, including clinicians and statisticians, and that can also be agreed upon by the review committees or the safety committees, and then can be used by the operation teams in practice to carry out the decisions. And therefore, it becomes almost like a 3+3 designs, because then all you need is to print out these tables and carry out the corresponding decisions. And the tables are functions of the target probability of toxicity, the definition of the equivalence intervals, and then sample size. Given these input, then the MTPI inference or MTPI2 inference or many new designs nowadays can generate a tables like this, which greatly facilitate the trial conduct and trial design. So in summary, since 2017, the contribution of these so-called interval-based designs is to be able to use statistical inference, model-based inference to actually generate rule like tables like this, and this seems to enjoy the both the nice properties from model-based inference as well as the rule-based design, such as 3+3, which has this type of very nice tables for practical usage. So other types of designs, such as cumulative cohort designs, the Bayesian design, I3+3 design, they further simplify the interval ideas, but they all provide decision tables like this. So I'll skip some of these slides and just to go to a last bit on designs on this table. So you can see that first we have the CRM, which are complex designs, then the MTPIs, which try to simplify the models and using up and down designs. Then the CCD and the Bayesian designs even further simplify the idea. What they did is they decided to perform inference based on point estimate rather than interval estimate from the probability model. So let's use Bayesian design as an example. It becomes very popular recently in the society. This is the typical, this is the main decision rules from the Bayesian design. Assume at a given dose, you treat number of patients, denote that as n, and then out of these n patients, you would probably observe y of them, y patients having DLTs. So you get two numbers, n and y. Y is less than or equal to n. And so you just calculate the fraction y divided by n, which is the empirical toxicity rate at a given dose. And then the rules are very simple. You compare this fraction, the observed toxicity rate, to the boundaries of the equivalence interval, right? So you compare if it's less than the lower bound of the equivalence interval, or if it's between the equivalence interval, or if it's greater than the upper bound of the equivalence interval, then you take the up and down decisions. So for example, if you're less than the lower bound of the equivalence interval, it means it's underdosing, and therefore the decision should to escalate. If it's in the equivalence interval, then you should stay. If it's above the equivalence interval, you should de-escalate. Very intuitive, right? And this type of rules was actually originated by the CCD design back in 2007. And both designs, so the Bayesian designs use the same framework as the CCD designs that apply the safety rules that were proposed in the early MTPI designs. So as an example, if the target is 0.3 and the equivalence interval is 0.25 to 0.35, then based on 3 patients, if you have 0 DLTs, then apply this rule, you would escalate. If it's 1 out of 3, you would stay. 2 out of 3, 3 out of 3, you would de-escalate. And similarly for when you have 6 patients, or you can have any number of patients, just apply the rules, right? So anyone can do it. Even the clinicians can apply this type of simple mathematics to generate the decision rules. So that's why it's becoming very popular. So the difference between the CCD design and the Bayesian design is the definition of the equivalence intervals. The O here stands for optimal equivalence interval, where there is a mathematical optimization behind the scene to optimize the original proposed equivalence interval under the Bayesian design. So in a paper we just submitted, we show that actually the MTPI2 Boeing and the CCD design are under the same decision framework. So if you know a little bit about the optimal decision theory, the theory says that given data, you have a probability model, and then there's an action. The actions are, for example, de-escalate, stay, and escalate. You write down a loss function. Then a decision rule is essentially a function that maps the data to the action. And then you can ask for optimal decision rules. Sorry, not optional, optimal decision rules, where it provides the smallest loss. The loss can be expected loss or some kind of loss. For example, if you want to use Bayes rule, then it minimizes the expected posterior expected loss. So these designs, the MTPI, MTPI2 keyboard, and Boeing, they are all based on the Bayes rule under the same models for the 0-1 loss. So in other words, they're all Bayes rules under the same set of decision framework. The only difference is the priors that are being used. For the MTPIs, the priors are truncated beta priors. For the Boeing design and CCD design, the priors are point mass at 3 points. The 3 points are the boundaries of the equivalence interval and the target probability of toxicity. So that's all. Otherwise, the 4 designs are essentially the same. They are all optimal using the Bayes rule. So the question becomes, which priors would you prefer? Would you prefer truncated beta prior intervals, or would you prefer point mass priors for under the CCD and Boeing? To me, I would prefer the truncated interval designs, because the point mass prior essentially are telling that I only believe that the probability of toxicity for the current dose takes 3 points, although it's a probability, right? I would rather not assume it takes only 3 points. I would assume it takes some probability distributions between 0 and 1 interval. So that's a little bit of a theoretical insight. And if you heard about this, the Boeing design recently get the FDA fit for purpose designation. In the FDA document, it pointed out that there are some changes needed for the FFP, the fit for purpose that's designated Boeing designs. So just to be aware that when you apply Boeing designs in the future, you want to make sure you're sticking to the proposed changes based on the FDA reviews. Otherwise, if you just use original Boeing design, there might be some questions. So these are some minor details on the Boeing design. So another really cool benefits of these simplified model-based designs are that sometimes you may not even need to run simulations to evaluate them, because all of them provide the decision tables like this. So what you can do is you can just compare their decisions and to see, which decision table is more sensible to me or to this trial. The reason is that when you run simulations, the simulation results is a deterministic function of 2 things. One is the decision table like this, and the other one are the safety rules and MTD selection rules. The designs listed here, they actually use exactly the same safety rules, MTD selection rules, and the only difference being the difference in the decision tables across the designs. So in other words, you can run simulations on different scenarios and compare these designs, and you can say, okay, out of these scenarios, one design seems to be better than the others. But the real underlying truth is that the only difference between these designs are the decision tables, because the other rules are the same. So you actually get more information by listing their decision tables and compare them side by side. So for example, we compared the in this case, the Boeing design and the modified CCD design, which it means the original CCD designs plus exactly the same safety rules and MTD selection rules, and we compare the differences. We table the differences in their decision tables for a range of target probability MTDs and then different equivalence intervals. So 0 means there's no difference between their decision tables, right? And this is, you see all zeros across all the epsilons, which are the equivalence interval boundaries. So you don't have to run simulations anymore. You know that these two designs will give you identical decisions, and they use the same safety rules. So anything you see any difference you see in the operating characteristics from the simulations are potentially due to the number of random seeds or any random errors in the software that are using. So, but essentially the two designs should be identical. So that's one thing I want to point out. And the other thing is, be careful of just looking at the simulation results out of a selected number of scenarios. This includes your own simulations as well as the simulation results that have been published in the papers, because to fully assess the performance of a design, ideally or theoretically, you want to run the simulations across an infinite number of scenarios, because you don't know what the truth is. So, but in reality, we only report the results out of a finite number of scenarios. So the simulation results sometimes can be misleading. Again, thanks to the simplicity of these designs, one way to augment the assessment of the performance of these designs is to look at the differences in their decision tables rather than just being completely focused onto the simulation results that have been reported or have been conducted. So be careful when you review the simulation results. So at the end, I'll introduce another new design and set up the stage for the novel development under the dose optimization. So this new design is called the I3+3 design. It's not super new. It was first submitted in 2019 and published in 2020. It's a rule-based design. So, and the motivation was actually from the CCD and the Boeing, where I mentioned briefly that you look at the framework under the two designs, they only use Y/N in their decision rules. Y/N, as we know, is the empirical observed rate, or the MLE for the binomial distribution, or the posterior mode for the binomial distribution and the uniform prior. So it's a point estimate rather than interval inference. And so with the point estimate, one can develop such powerful designs like these two. Then this motivated me a few years ago to think about, remember, why do we use probability model statistics for inferences? Because we want to account for variability. But the point estimate does not have variability. Therefore, I asked the impossible question, which is, can we actually develop a new design which stay away from models? And remember, this was the whole motivation of improving the 3+3 design 30 years ago, which is to use probability models to enhance the design and performance, right? Now it seems like we're going backward, and I call this a spiral evolution. We're considering abandoning the models and using rule-based designs, at least for phase 1 trials. And the answer is yes, there is such a design, and the design is the I3+3. So the I3+3 now abandons all those probability models. It tells you a pre-specified set of rules, just as the 3+3 design did. And the rules follow this very similar framework as in the CCD and Boeing design, where you compare the observed toxicity rate with the boundaries of the equivalence interval. So if it's less than the left boundary, lower bound of the equivalence interval, it means underdosing, you escalate. If it's inside the interval, you stay. If it's above the interval, this is where the tiny little bit innovation comes in. it asks you to look at 1 minus 1 over n, meaning you take out one DLTs from the observed data and then recalculate the observed rate. If that rate is actually less than the lower bound of the equivalence interval, then you have a situation where a unit change in the observed outcome resulted in from going above the equivalence interval to below the equivalence interval. In other words, the information in the observed data is very sparse. So even though you observe a rate that's above the interval, it's probably associated with high variabilities, and therefore the decision is to stay rather than to de-escalate. If this condition is not satisfied, then the rule believes that there's enough variabilities in the data to back up this greater than the upper boundary overdosing statement. Therefore, you should de-escalate. And that's it. These are the 4 rules. And if you apply these rules, you get a new design. And we call this the I3+3 design. It's an IA plus B design because you don't have to do 3 plus 3. You can do any A plus B because all you need is to get Y and N from that dose. So this is just an example of what the rules are under the I3+3 design. You could, for example, just apply the I3+3 rules. You'll get 3 patients and 0 DLTs. You would escalate in the MTPI and 2 and Boyne, you would do the same thing. But when it's 1 out of 3, then the I3+3 design would to stay. Under the 2 designs here, it would de-escalate. And this is under the target of 0.3 and the equivalence interval of 0.25 and 0.33. Recently, regulators seem to not like having equivalence upper bound more than 1/3. And therefore, we are considering a case where the upper bound of the equivalence interval is 0.33. So you can see the differences here. The I3+3 design would to stay even under this setting, but the Boyne or the MTPI2 would to de-escalate. Which one do you like? I don't know. I think either one could be reasonable. If you're asking clinicians, they are probably more used to stay the decision here because they're used to the 3+3 design. That's all. But theoretically, you can show actually the I3+3 CD and Boyne have identical decisions when the sample size at a given dose is large enough. As long as 1 over n, the sample size at a given dose is less than the length of the equivalence interval, then the 3 designs give you identical decisions. So the only differences between these 3 designs is when the sample size is small. So I skip this just to demonstrate, this slide is just to demonstrate further differences between the 3 designs. So as I mentioned, how do we assess the performance of the design? Because they all give the same safety rules and MTD selection rules, the only differences in their simulation would be driven by the differences in the decision tables. So again, we did the same thing comparing the decision tables of the Boyne and I3+3 design under different equivalence intervals. This is out of 1,326 decisions. So you can see the largest difference in the decision is 14. That's about 1% of the entire decisions we compared. In other words, these 2 designs should perform very similarly because their decision tables are very, very similar. In many cases, there's no difference in their decision tables. So I think I will skip this a little bit. This is just to motivate or to illustrate when you actually compare different designs in simulations, there are a lot of metrics that you can summarize to compare. So for example, you can compare the probability of selecting the right MTD, the allocation of the patients, the overall toxicity of the trial under the design, when the trials are stopped, the trial sample size. Each one of the metrics tells you some part of the story in terms of the performance. However, it's very hard to get an overall assessment of the designs because at least here we have 5-dimensional metrics. How do we compare across 5-dimensional subject? We don't know how to do that if we just want to use all 5 of them individually. However, one could summarize them into utilities. So this is a work we're still working on. It's called the J-score. we want to combine these different metrics into a single utility value, and therefore you can calculate them. We did some preliminary simulations to compare these designs using the simple utility score under different scenarios and using different drug developmental strategies. The P1, these P's are the strategies or the programs, I think. The P1 and P2 means that the strategy is to escalate as fast as possible. It's an aggressive strategy because there's a prior knowledge that the drug or the therapeutics might be very safe. P3 is a neutral one, and P4 and P5 are conservative ones where the drug or therapeutics might be considered to be potentially toxic at higher doses, and therefore you want to be careful in those escalation decisions. So under different programs for different types of scenarios, these scenarios are arranged in terms of the difficulty, like a effect size between the adjacent doses. And then we assess the multiple designs in terms of their utility scores. And we listed the top 3 performing designs under each one of the program for each type of scenarios. You can see the top performing ones. There seems to be a pretty interesting results, and was also very surprising to me that there are a lot of I3+3s on the top tier. But in general, you can see different designs show up in different places, which means all of these designs are performing pretty well. And then no designs dominates another designs in all cases, as we know that there's no such thing that one model will be better than another model in all the scenarios. That's just not possible. So to summarize, for these classical dose finding designs aiming for MTD, there are many, many good designs out there now. So for non-statisticians or for practitioners who are less concerned about the statistical theory, but more about the easy to use practical operations, then the I3+3 Bayesian CCD design seems to be really, really easy to implement and with good performance. For statisticians who care about the balance between using model-based inference, using intervals, and accounting for variabilities in your decision rules, then these MTPIs or keyboard, the semi-parametric model framework may be considered. For hardcore statisticians who really want to use model-based and principled inference, such as borrowing information across doses, then the CRM, BRM, or the overdose escalation with overdose control can be considered. So there, again, there's no one-size-fits-all decisions here. It's all about the personal preferences. So this is a quick summary, and I won't go into the details because I want to spend some time on the new era. So in general, the modern oncology phase 1 or early phase development are becoming more sophisticated and more powerful. So for example, we no longer just conduct a phase 1 trial to find the MTD and then do something later to figure out the phase 2 proof of concept. There are many seamless phase 1A dose finding plus phase 1B cohort expansion. I think FDA just released the guidance on expansion cohort studies. So one can design a single trial that combine phase 1 and phase 1B together. And then in particular in the cohort expansion, sometimes in oncology, we get a basket trial where we can apply Bayesian hierarchical models to borrow information across different indications of doses. For the immune and targeted therapies, MTD may no longer be the optimal dose. Remember in the picture I showed you there, the reason we want to find MTD is the among all the safe doses, the best one in terms of inducing efficacy. But when the dose response curve for efficacy is no longer monotone, then MTD may not be the optimal dose. And sometimes you may have multiple candidate doses for expansion due to the dose response or exposure relationships. And then sometimes a drug can potentially work in multiple cancer types, such as non-small cell lung cancer, et cetera, et cetera. And also there sometimes you get delayed outcomes, right? So after DLT is defined, is an event defined for the first cycle. In immune, for example, some immune oncology drugs, the toxicity only show up later in the cycle 2 or 3. So how do you accommodate that? And also we are into the era of combination therapies, right? Sometimes we would even run into novel combination, although there are not many, but still, how do you deal with those findings for combination? And if the efficacy is no longer monotone, then a natural statistical remedy is to consider the joint outcome of efficacy and toxicity, for example, in those findings, like CAR T, gene cell therapies. How do you accommodate that? And the other thing is, can we speed up the trial enrollment by allowing rolling designs, especially in the presence of delayed outcome? If you want to wait for 2 or 3 cycles to observe the toxicity outcome for every cohort, the trial could be prohibitively too long. And therefore, some innovations have been proposed to speed up the enrollment. And last but not the least is nowadays there's multi-regional drug development. So maybe phase 1 is launched in some part of the world, and then later another phase 1 trial is launched in another part of the world by using the same therapeutics, right? So ideally, you want to borrow information even for those findings. How do you do that? So let's set the stage for dose optimization. This was a recent, very famous case, I think from Amgen, sotorasib for non-small cell lung cancer. It's already been approved here for patients with non-small cell lung cancer harboring the KRAS mutation. This is the first drug that successfully targeted KRAS. KRAS is a very well-known cancer biomarkers, but they historically has been considered as undruggable because of the biology. I know a little bit about it, but not good enough to present my knowledge here. But it was traditionally considered undruggable, but now we have a drug, which is great. However, a post-marketing trial was required by the FDA to further explore the lower doses than the approved one. And then potentially this is due to insufficient dose exploration in the early phase, such as small sample size phase 1 trial, which we all do, or dose selection under the MTD regime. So in the New England Journal of Medicine paper published by the FDA last year, they not only listed the sotorasib as an example, but motivating example, but many existing approved therapeutics as examples which require post-marketing drug dose exploration studies. These are quite expensive studies because they're done after the drug approved. And then the tables listed here compare the original approved doses versus the modified doses after the dose finding trials are complete, post-marketing dose finding trials are completed. You can see in almost all the cases, most cases here, the doses have been reduced, and the reason for reduction is all due to the toxicity. So in other words, we didn't do a good job for these drugs in determining their doses, mostly in the early phase trials. So now there is a shift in our focus, at least for novel oncology drugs, therapeutics. Instead of finding the MTD, we want to optimize the dose. And this is not new for non-oncology drug development. We know in non-oncology, phase 1 is on healthy volunteers. Phase 2 is the dose finding or dose ranging using randomization. But for oncology, traditionally, we've been just rushing through early phase finding MTD and go through proof of concept really quickly. And then, sometimes now we even prove drugs at the end of the phase 2. So because of the new innovation in biology, in developing these novel immune and targeted therapies, MTD is no longer the optimal dose. In other words, higher doses might not have better therapeutic activity. And then the DLT may not be observed at clinically active dose. Sometimes you run through the entire dose finding trials and you find no DLTs in all the doses, then what you do, right? And serious side effect may occur after several cycles of drug usage, and therefore you may need to deal with delayed outcomes, including delayed toxicity or even delayed efficacy. So as a consequence, MTD is no longer optimal. So the designs that I mentioned so far, they all aim to find the MTD. As you can see, if MTD is no longer our target, then all of those designs are no longer suitable for these new trials. So for example, if that's the case, then one could consider the joint outcome of efficacy and toxicity, or even some combination of biomarkers as surrogate efficacy outcomes with toxicity. And then you would want to also consider exposure data in dose finding trials, as well as modeling the time to toxicity rather than the binary outcomes of DLTs. And there are different ways of doing that. Either you use the time to event modeling, or we recently proposed a new framework called the probability of decision modeling for dealing with the delayed outcome. And so to start, I want to point out there was this paper we published many years ago now, 8 years ago. I was considered to be too advanced at that time. This is called the Sears design. It's a seamless phase 1/2 oncology dose optimization design, where the design proposed to run a phase 1 trial, a dose finding trial based on a design, any design you like. And the goal is still to find the MTD. And so where you imagine here, there are 5 doses, and the dose escalation is being conducted up and down these 5 doses. And then you could apply a graduation rule. This graduation rule can look at both toxicity and efficacy, or even exposure. And so when the graduation rule is applied, some doses may satisfy the rules, meaning they would be considered as efficaciously promising. Toxicity-wise, they are safe, and exposure-wise, probably good, meaning they're within the therapeutic window, they have sufficient exposure. Then you can graduate that dose into a phase 2 seamlessly. So what this means is, for example, D3 is taken out from the phase 1, while the phase 1 would continue on the remaining 4 doses. And then D3 goes to phase 2, and phase 2 could even have a control arm, and you can start a randomized controlled phase 2 trials comparing the efficacy, or even, I don't know, PFS of the D3 versus the control, okay? And so at this point, both phase 1 and phase 2 are ongoing at the same time. Later, you could imagine that the D4, another dose, could satisfy the graduation rule. Then the D4 would also be graduated into and join the phase 2. And at this point, there might already be some patients being allocated to the D3 and D0, the control, but that's okay. D3, D4 will join as in a platform type of trial setting. And then Bayesian adaptive randomization may be considered to mitigate this type of innovation. This is indeed what was proposed in the Sears design. And so the 2 trials will be ongoing at the same time. At the end of the entire study, you would probably get a optimal dose, which is D3, and this D3 could become the recommended phase 3 dose. And so in other words, if you run these 2 trials, then it's likely that you get a dose, and that dose could be directly pushed to the phase 3 studies. So this was proposed 8 years ago, and was considered probably too new at that time, because at least there was no initiative such as dose optimization as we're talking about here. But it seems to be quite relevant now, especially that we don't really want to just use phase 1 to find the MTD, and we want to learn about not just the toxicity, but the efficacy of these doses as efficient as possible. So another innovation that we considered was to borrow historical data or external data, trial data in dose finding. And this is based on the I3+3 design. The new design is called the high 3+3 design. H stands for historical data. The design has recently been applied to a couple of trials already in two different settings. The first, in the same setting, actually, both trials, they already accumulated historical data. So as I mentioned, there was a case where the trial was already initiated in China with one drug, and then later in US, the sponsor wants to launch another dose-finding trial using the same drug, but during the IND profiling, there's already data from the Chinese studies in terms of the toxicity outcomes. So this is our Shiny app. And here you can see this is the design for the US trial where we have a target, the equivalence interval, number of doses. All these are not specific to that trial. Obviously, it's confidential information, but this is just an illustration of the R Shiny app. And the maximum number of patients per dose. We used a power prior which controls the effective sample size at every single dose. But the main innovation is here. It allows you to impute historical data from, in this case, the Chinese trial. And then at the moment, for example, again, these are fake data. Suppose the Chinese trial has a data with one patient at dose 1, no DLTs, 3 patients at dose 2, no DLTs, 3 patients at dose 3, 1 DLT, and no patients at dose 4 and 5, then the design would actually generate decision tables like this, but one for each dose. So remember in the previous interval-based designs, you get one decision table for all the doses. Here you get a one decision table for every single dose because it's trying to accommodate these different data observations at different doses. But still, it's simple enough because you can just print out these 5 tables and then use them to run the trial. So this seems to work pretty well. We've run many simulations comparing the performance, and it's much better than I3+ design without borrowing information, and it seems to be doing a reasonable job. So this is another extension of the I3+3 design. As you can see, I'm just introducing the things that we developed recently. By no means these are the best. There are many other development out there, but I'm mostly familiar with these designs, and I want to introduce them to the audience today. So the JI3+3, and J stands for joint outcome. So if you want to consider efficacy and toxicity for dose finding, especially let's say in cell and gene therapies for liquid cancer, because in those indications, the efficacy can be quickly observed, and therefore you can use the joint outcome for dose finding. Then the idea is to extend the I3+3 rules where on the horizontal axis here, we have the equivalence intervals for the toxicity, the underdosing interval, and the overdose interval. On the efficacy scale here, we only have a reference or target efficacy rate, but we can apply the same principle of allowing the observed toxicity rate and the observed efficacy rate to drive the up and down decisions. So remember in the I3+3, if this observed rate is less than the equivalence interval in the equivalence interval, then we would do up and down decisions. And similarly, if the observed efficacy rate is below the target, then we want to escalate, right? That's logic. If it's above the target, we don't want to escalate. We want to stay there. The innovation or the borrow of innovation in I3+3 design is to consider Y-1. Remember, taking one DLT out of the data and recalculate the observed rate. When depending on how big this value is, if it's less than the equivalence interval, then remember we would then to stay rather than to de-escalate because of the sparsity in the data. So we apply the same philosophy in this two-dimensional decision table or picture here. and coming up with these decisions. So if you're here, means both efficacy and toxicity are under dosing, then we want to escalate. If you're here, you're either de-escalate because the toxicity is too high, or stay if y minus 1 over n is less than the equivalence interval boundary, means there's not enough data to support the high observed toxicity rate, and then the decision would be to stay. So you can see applying these 6 this up and down decisions across the regions of the both efficacy and toxicity probabilities, one can achieve the same simplicity in applying the I3 plus 3 for toxicity only dose finding. Then if we want to deal with delayed outcome, it becomes a little bit more complex. We have to model the time to event toxicity. The idea is that when you enroll a cohort of patients and during the follow-up period, if you have a new patient that is eligible for enrollment, traditional design would turn them away because you want to wait until all the patients in the cohort have completed the follow-up here, okay? So before this box here, you would not be able to enroll any patients. The design would to be able to allow you to enroll at least at some time based on the time to event data. So the fact that, for example, this patient has no DLT up until this point here is information, right? It tells you potential something about the toxicity profile for that patient. So there are 2 options to do it. One is to consider this as a time to event data. The other one is to consider as a missing data. And there are many existing papers out there, either using the survival likelihood or augmented likelihood. And we show in a paper published 2 years ago that these 2 types of approach actually are identical. And oops, my froze. Okay. However, we proposed the new framework where we take a step forward. We want to calculate the probability of decision. Remember, the term probability of success is essentially a type of Bayesian posterior predictive probability. Same thing here. The probability of decision is a posterior predictive probability of decisions by accounting for these time to event outcome. I think I'm missing 1 slide here, but it's okay. The idea is to model either using the survival likelihood or augmented likelihood, and then impute the missing data and based on posterior predictive probability distributions, and then apply, for example, MTPI or I3 plus 3 type of rules for the imputed data. And by imputing that many times, you can actually get the posterior probability of the decisions at the end of imputation, and then use those probabilities to generate up and down decision rules. So part TPI, probability of decision TPI, is such a design that applies the type of inference. This has been shown, at least in our simulations, to reduce the trial duration by quite a bit, but still be able to maintain the safety. One issue you have to be aware of is if you want to allow enrollment here and make a decision on the dose for that new patient, you are under the risk of making a wrong decision because it's perceivable that the missing patient may end up having a lot of toxicity later on. So there's always a risk for making these early decisions. And so the probability of decision framework actually allow you to quantify such risks and therefore be able to mitigate that risk. This is a major benefit of the POT framework when compared to other designs like a tight designs, tight CRM, for example. So I'll end here. And then, so in conclusion, as you can see, although the dose optimization seems to be a relatively new concept, the Statistical Society has made a lot of innovations already considering these challenges for dose optimization. Most of these designs are still, they're very new. There haven't been many use cases in practice, so they need to be tailored and in collaboration with pharmaceutical sponsors to improve the conduct and the implementation of these designs so that the logistic requirements are met. We know that papers, normally they cannot directly be applied to practical trials. So there's needs to be collaborations and case studies going on to refine these designs. But because of these innovations, I believe the oncology early phase trials are getting more efficient, although they're becoming more complex, but that's okay. And because these innovation, they are benefiting the drug development to make it more efficient, faster, safer, and more powerful in identifying the right doses, and therefore benefit the patients at the end. So I'll stop here and thank you for your attention and happy to take any questions.
Yeah, thank you, Dr. G. This was a great presentation. Any questions? Please feel free to unmute yourself. There is one question on the chat.
Okay. Sorry, well, chat.
Yeah.
For some reason, I can't see the question.
Oh, sorry.
Yeah.
I think that was directed to me, but yeah, I can pose the question for you. The question is, should sponsors, for example, CAR-T therapies contact you to implement some of these, and how? I think it's a more of a pointed towards you, like a personal question, I think.
Oh, of course, I'm happy to help out. Yes, the answer is yes, I'm happy to help. There are other choices too. So I'm pretty sure, you know, there are other experts in the field that can help out. But for CAR-T, I'm very happy to help out. The JI3+3 design was developed specifically having CAR-T therapeutics in mind. And then I think it's actually necessary for CAR-T trials to consider the non-monotone relationship between the efficacy outcome and the dose. So yes, happy to.
Yeah, thank you. This is John Constant. I posted that question. I work in a very large CRO and have recently come across several sponsors using CAR-T and TIL therapies, and these particular sponsors are very innovative minded and want to do something more advanced than you know, modified 3+3. So I may contact you about those in the future. Thank you.
Of course, thank you.
Dr. G, this is Frida Kuhner. I really appreciate your presentation. It's a very practical tutorial of all those models. In real life, I do have a question on dose escalation portion that we usually don't do a simple dose escalation. It's more of a dose exploration, including exploring different frequencies or different step dosings. I wonder if those models can be adapted to accommodate those kind of dose exploration exercise.
So can you let me know a little bit more about the dose exploration? What do you mean by that? Do you mean the actual doses are not pre-specified, or?
The doses are specified, but it's more specified in the fashion of a target dose. So during the dose escalation, we will figure out maybe if we start with the lower dose and eventually escalate to the higher dose in the same regimen, you will mitigate some of the safety issues. So those step dosing actually has a causal relationship of the DLT we observe. However, in those the models you explained today, I don't know if there is a very straightforward way to incorporate that kind of information in dose finding.
So first of all, I'm still not clear, but I think if the decisions for dose escalation are based on the observed toxicity, or observed time to toxicity, or observed toxicity and efficacy, then the designs that are introduced today can be applied. So in other words, for example, if you're only considering toxicity, and if a dose is shown to have no DLTs, right, out of the treated patients, then the decision would be to escalate. And if that's what the trial does, then these designs like MTPI 0.93+3 can be applied. Does that make sense? Or
Yeah, I think so. I understand that it's if we think about a regimen as one dose, eventually all those models can be simply applied. I think it's more of a consideration, say, if we start dose 1, then the second dose is dose 2, and the third dose is dose 3. So this is one regimen, and the other one is we just start with dose 2, and the second dose is dose 3. That's the first cycle. We use that kind of regimen. I'm not sure if I can use the model to give that kind of recommendation, which one is safer, or what should be the next dose to consider. I'm not sure if I explain myself very clearly.
oh, so when you say regimen, then this ring a bell in my mind. So do you also include the dosage and the frequency of the schedule?
Right, we do have QW once weekly,
Yeah.
yeah, the Q2W kind of consideration. Once we have add all those noise in the dose finding, dose selection, it's a little complex to simply apply those models.
Yes, agreed. So then now I understand. So there are, if you treat schedule as another dimension, then I believe there are designs out there that consider both dose and schedule. You can consider them as like a two-dimensional dose finding design, where they want to optimize the regimen, which is, I think, what you are referring to. So in that case, I didn't touch that part of the designs today in the slides today. It's a separate topic. Yeah.
Great. Maybe we can have a follow-up presentation. Yeah. Thank you so much.
Sure.
Hi, this is John Lowy. I'd like to ask you a question about the previous data that you might use, because I find, especially in oncology, it's hard to put together those patients who have failed multiple treatments prior to coming into this new study versus those patients who are maybe fresh. How do you sort of either separate those people out or quantify that in preparing for making the comparison, if you understand what I'm trying to say?
Yeah, I do. I think it's a complex question.
Well, in a sense, how did you divide out the data when you're looking at the prior data for making a comparison for your proposed studies? Do you use clinicians to help you pick out the patients that are the most comparable? Because to me, a lot of this modeling is really dominated by the patient variability where they are in their treatment.
Yeah. So I can give you some of my limited experience. One, I think in certain parts of the protocol or in certain trials, there is a specific like exclusion criteria, right? Trying to enroll relatively homogeneous patients. When that's not the case, I've also seen things get very complex. Right? In addition to what you mentioned, there are also even say for patients who have similar previous treatment, they could be treated at different places. The protocols for those treatment could be different. They could be treated at different regions, right? And so what I've encountered is a lot of times physicians have some kind of internal assessment in their head. In terms of statistical modeling, I haven't seen any designs or methods taking that into account. For potentially one reason, the sample size for these early phase trials are typically very small. So there is usually not enough statistical power to capture that. When you say the patient have different history, that could mean a lot of things, right? It could have multiple variables that are needed to describe those different histories. And if you only have like 6 or 3 or 12 patients, it becomes very challenging to capture those subgroup effects. So unfortunately, I think in early phase trial development, at least, these factors are usually not explicitly modeled in statistical methods.
Yes, and I'm just a quick follow-up. How do you, because you mentioned biomarkers, especially something like CRAS, how do you take into account the prevalence of the biomarker if it's relatively rare when you're making the exploration? Because it's hard to, again, to group people together then in that regard.
Yeah.
I wanted your thoughts on that.
Yeah. So I don't want to sound like I'm just advertising all the methods we develop. I didn't put it in the slides. We encountered that there has been a new design. We haven't published it, where we allowed, in those findings, we allow the main cohorts to be able to enroll patients of all comers, right? Usually in phase 1 oncology, we don't really care which patients, or in terms of biomarkers, or even sometimes their cancer types. But we allow backfilling of specific targeted subgroup, like a KRAS mutated subgroup, which could be less frequent, right? So once we clear a dose on the main cohort to be safe, the main dose finding cohort goes to the higher dose. But if, for example, the PK indicates that we're into the therapeutic window, then a backfilled cohort would be allowed to enroll specifically on the subgroup, on the for example, KRAS mutated patients. That could help. Yeah, that could help. But again, we can't speed up. We can't just speed up the enrollment of KRAS patients if there are very few of them, right? We just have to, be patient and wait for them to show up. But at least the design allows you to enrich such population in those findings.
Thank you very much.
Sure.
Great presentation.
Thank you.
Any more questions for Dr. G? All right, I'm not seeing any. Don't see anything on the chat as well. So if there are no more questions, many thanks to Dr. G for such a clear presentation, an interesting presentation, and would like to thank you all for joining the session today. Have a great weekend. Yeah.
Thank you.
Thank you, Dr. G. Bye.
Thank you. Bye.